And speaking of which, the first word. So for Cook, who's here, and Burton, who's online, and Hannah, who's online, these will be extra credit words. But for anybody watching the video at a later date, this is just one of the words. But anyway, actually, let me wait. We got one more person coming in. All right. So the first word it will be word. So the word is the first word. Word. All right. And again, for those of you who are here live, that's extra credit or extra credit for you guys. And if you're watching the video at a later date, that's just one of the required words is word. Hope everybody did good on the exam. We'll see you soon. The scores were meant not to be released until the exam was closed. Because the people before you have cheated. So one of the ways to prevent that, or to help prevent it, is to not give anybody their score. So if John takes his exam and scores a 98%, then obviously when his buddy goes to take his exam and they're the cheating type, they might say, "Hey, John, why don't you help me with this exam? Because we know you got a 98%. That's the kind of thing I'm trying to avoid. Actually, how many of you, can I get your opinion? I know there's only a few of you, but did the whole black word, red word throw you off too much? I tried to make it look like the black words were just kind of a blur. You could just red words, red words. Was that okay for you guys? Okay. There's an app where if you take a picture of a question, it'll just give you the answer. So doing that helps combat that. You can still use that app if you know how, and if you have the time to manipulate it. But with 50 minutes, you don't have the time to manipulate it for every question. In case you're wondering about the method behind my madness. All right. So we have another person online. So we'll do another extra credit word. And it is extra. Actually, now we have two more people online. So yeah, that word is extra. So again, if you're online right now, if you're in person right now, that will be an extra credit word. And if you're watching this video at a later date, that word extra is just one of the required words. Anyway, let's see. We've got Ho not here. It might be easier to just do who's here. Cook and Red is physically here. Burton is online. Hannah is online. Kincaid is online. Hughes is online. And Durgan is online. And Hunter is here. Some other reminders. Let's see. By Sunday, you should have 80 points of independent work. Speaking of independent work, I'm sure some of you have noticed I am very picky, especially the longer the semester goes on, the pickier I am. Because in theory, you would have had some time by now, 7 weeks, 8 if you include spring break, to start turning things in and seeing how I grade. And plus, I've been reminding you guys of a lot of things throughout the semester. So at this point, I'm a little bit pickier. To that point, I think the biggest problem I'm seeing is the problem with sources. So make sure you guys use your sources and use them properly. Specifically, the biggest thing I'm seeing is information in a paper that's not in a source. And I hate to do it for you guys because I know genuinely probably some of you just did it on accident. But because AI is such a thing now, everything has to be accounted for. Every little fact in your paper has to be accounted for. Because I've tested AI and it does a really good job of a lot of things. But what it doesn't do a good job of, even when you ask it to, is cite sources. It'll look good, it'll cite sources, and if you just briefly look at your paper or the paper that AI wrote for you and briefly look at the resources it provided you as the sources, it looks like it's legit. But if you're like me and you're grading it and you're really digging in and you're looking at every single fact and comparing against the sources, then I'm going to find mistakes, right? And usually I think I hope for your sakes, it's your mistake and you're not cheating. But I didn't want to use the word cheating. So AI is a tool. So again, I'm going to say officially it is against the rules to use AI, but unofficially it is a tool. So if you feel like using AI to write yourself a paper, then you do what I do when I grade it before you turn it into me. Check it thoroughly. You look at every single fact to make sure it's accounted for in those sources that AI gave you. Or if for some reason you didn't even ask AI to give you some sources and you're just looking some sources up, look them up carefully, please. It's a weird thing, this AI. It really is a good tool. And I think it's just so early, it's hard for me and a lot of other educators to come up with rules. It's almost tantamount to saying when Google first came out to say, you can't use Google, right? So I almost feel bad for saying you can't use AI, but that's only because I'm still figuring out how I want to run things. So again, officially, I'm saying don't use AI. Unofficially, I'm saying if you use it, you better be very, very careful. Because in my opinion, too, the purpose here is for you to learn, right? So if you write a paper or you have AI write you a paper, and then you go through it fact by fact by fact, you're learning something. You may have not written a paper, but you're going to learn something if you read everything and make sure it's all right, the paper says this. Now I've got to go through this whole source and find where it says that. Does that make sense? So to me, in my opinion, using AI properly is actually harder than writing a paper. So just write your papers. Don't use AI. If you do use AI, check your sources a little bit more, well, not a little bit more, just very thoroughly, 100%. What else? And also the other biggest problem I've seen with independent work, and this is really hard, I understand. Don't reword your sources, right? So your paper shouldn't look like the same thing, your source, just with different words, right? A lot of the stuff I'm seeing is sentence for sentence. It's the same thing as your paper, as your source. You just change some words. Instead of saying the dog quickly ran to its mother, you might say the dog very quickly ran to its mother. No, you can't just throw in some extra adjectives or whatever. And again, I'm not trying to be picky. The point here is for you to learn. That being said, a really short paper, I will be a little bit more lenient on that. It's kind of hard not to, if you're just answering a question and you have a source and you have a sentence or two, then that kind of makes sense. How are you going to reword a sentence or two? I was wondering why I'm 6'6" and I have size 15 shoe, and my brother is 6'9" and has a size 13 shoe. Is there a genetic component to that? I looked it up, and according to this source, blah, 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 right? And then if that's it, that's the extent of your paper, obviously you're going to have a short paper, and obviously it'll be a little bit harder to summarize it in your own words, if that makes sense. Because with just such a small amount of information, sure, you might just have the one sentence from your source that says the one thing you're trying to tell me. I hope that makes sense. Anyway, we've got some more people, and it's 5 minutes early, so let's give some extra credit, especially for those people who are online who don't usually get extra credit. So again, this is not a required word unless you're watching this video at a later date. So if you're here in person, or if you're here watching this live online, the first word for attendance, or this extra credit word for attendance, will be red, like the color red. So anyway, that's the announcements and reminders. Anybody have any questions about anything? Now that exam 2 is over, well, technically it's over at 9 a.m. But anyway, once that's over, it is then too late to retake the first exam. Well, once you've taken the second exam, it's too late to take the first exam. So that being said, now that the first exam is completely behind us and you can't even go back and make any corrections, if you meet me online and cover, so we can go over your first exam and I can tell you what's wrong, what's right, why the wrong questions or why the wrong answers are wrong, or why the, excuse me, I can't talk, why the correct answers are correct, then you'll get your 5% boost to your grade. Unless, of course, you had 100%, because, you know, what am I going to tell you? If they're all right, I'm not going to give you a 5% boost for that. So anyway, yes, now that exam 1 is behind us, you can meet me online so I can give you all the correct answers for exam 1, which you'll need because I'm going to use those questions to write the final exam. So I highly recommend it. What else? I guess that's it. If you guys think of anything, let me know. There's a lot of people online today, must be because it's a Friday. I would do the same thing. All right, let's see. Actually, let me do this first. So, Kincaid is online. I already said that. Andrea is online. Fisher is online. And again, just because I'm calling your name, if you're online, that doesn't necessarily mean anything. For points, it means nothing. Make sure you send me the words after we finish. Lewis is online. Cooper is online. Caleb is online. Crowder is online. Julia is online. Hannah's online. Hughes is online. And Burton is online twice. Morning. So now I'm going to take attendance for everyone else. So if you are online, if I don't say your name, don't worry about it, because again, you're going to need to send me the words. So uh-oh. She says she can't hear me. Still can't hear me? No one can hear me?
I can hear you.
Okay. Thank you. Perfect, guys. Thanks. All right. Less than a minute. Yeah, let's do some attendance here. So again, if you're online and I say you're not here, don't worry about it. Just send me the words at the end. You can just ignore when I say not here. That just means you're not physically here. Anyway, Abigail is here, physically here. And again, Kincaid is online. Andrea is online. Mullins is here. Lewis is online. Cooper's online. Hunter is here. Melton's online. Caleb, Melton's online. Crowder's online. Reinhardt is not here. Isabel is physically here. Julia is online. Talbert. Kennedy is here in red. Lecture credit. Hannah is online. Larissa is here. Madeline is here. Hughes is online. Galloway is not here. Cook is here in red. Durgan is online. Tate is not here. Burton is online. Kinder is not here. Jones is not here. Canterbury is not here. Hobson is here. And Fisher is online. Anybody in person that I missed? It's 8:01. So for those of you who just came in, if you have any questions about exams or independent work, then when I put this video out later, you should watch it, at least the beginning video, because I gave some reminders and some hints about exams and independent work. So watch that part of the video if you want. But let's get started. Well, does anybody have any questions before we get started? All right. Here we go. Let's finish this off and start the weekend. So again, let me back up a little bit. What we're talking about here is variations in Mendel's law, right? So in other words, well, I kind of hinted at this when we were talking. Actually, sorry. Let me do the word first. The first word for attendance will be hint. H-I-N-T, hint. Anyway, I was hinting around at this during lab when we were talking about, chin dimples and eye color and widow's peak, all that stuff. First of all, it's really unimportant in the grand scheme of biology. Like, you don't need to know that having a widow's peak is dominant. That's useless information. Those examples were just used as a way to give you some experience doing Punnett squares and talking about dominant versus recessive and things like that. Another thing that I kind of hinted at, and I'm telling you now, is that that's also very rare and oversimplified. In some of those cases, it's not even 100% true. So the eye thing was definitely oversimplified. The chin thing was not oversimplified, but the point I'm trying to make here, that it is one of those very rare things. Very rarely is there a trait or a character that is controlled by only one gene, and it's simply dominant versus recessive. So we start this conversation. We had this discussion, this lecture, talking about those things because it's the easiest to talk about. It's very simple. There's just one gene. There's two different alleles. It's either this or that, right? Pretty simple. But now what I'm telling you is, as simple as that is, it's really not normally the case. So what I'm about to teach you now is more realistic. These are the exceptions to the things I've just taught you. And in a sense, it's more important because it's more, I don't want to say realistic, but more often the case. But sadly, it is also a little bit more complicated. But let's talk about it. So again, the first thing what you need to know so far is the stuff we've already talked about, which is that there's dominant and recessive, right? When something is dominant, as long as you have one dominant allele, you have that dominant phenotype. To have a recessive phenotype or disorder, you have to have two recessive alleles, right? So that's what you know so far. Let's talk about the exceptions to the rule, right? So this is all the stuff. This slide's basically what I just told you, but in different words, right? So the first thing we're going to talk about is incomplete dominance. So that being said, let's give a word to what I've taught you before, and that word is complete dominance. So if you get an exam and I say, someone with a dimpled chin made babies with somebody with a smooth chin, and all the offspring had dimpled chins, what is this an example of? And you're like, oh, that's complete dominance. So that's a lot of the questions on the exams are going to go like this, where I'm going to describe a situation hopefully that you haven't heard of. Maybe it's even hypothetical, like a completely made up situation. But when I tell you this thing made it with this thing, and these were the results, you should be able to pick out whether it's complete dominance, incomplete dominance, which I'm about to teach you, codominance, biotropy, polygenetic inheritance, or epigenetics, right? So that's how the multiple choice portion of that exam is going to go. So let's talk about incomplete dominance. Incomplete dominance, the way your book writes it, is in the F1 phenotype, or excuse me, the F1 phenotype is between those of the two parents. That's the official textbook way of saying it. A better way of saying it is, in a sense, the kids are a blend, truly a blend of the parents, right? So again, if your dad has a dimpled chin and your mom has a smooth chin, and you're born with a dimpled chin, that's not a blend of your two parents, right? You just got your dad's chin. Does that make sense? In this case, it truly is a blend. So some examples, you don't need to write these examples down because they're just examples. I'm not going to use them on the exam. A white flower crossed with a red flower, right? If we were talking about complete dominance like we were previously, then the flowers would all come out white or red, whatever. But in this case, it truly blends the two. So what happens when you mix white and red? You get pink, right? Or another hypothetical example, you have a black mouse bred with a white mouse. If it's incomplete dominance, then the offspring are going to be gray, right? They're going to be a blend of the two. a curly-haired dog with a straight-haired dog. If you mix the two, you get a wavy-haired dog, right? Once again, you're blending the phenotypes. Incomplete means you're blending the phenotypes. Does that make sense? Hope so. Very important concept. So again, the thing we talked about previously to this, the complete dominance, was that one dominant phenotype dominated over the recessive phenotype. And in this case, it's a blend, right? Because none of them are dominant, really. I mean, we'll still use this term dominant and recessive when we look at this, maybe sometimes. But the point is, none of them are completely dominant, right? So when you mix a red flower and a white flower, it's not like it's mostly white or mostly red. It's just straight up pink. So the only reason I'm saying this so much is because I'm hoping that helps you remember this for the exam. It's because both of the parental phenotypes are incompletely dominant. None of them are completely dominant. Anyway, any questions so far? All right, here's a visualization. This is from your last textbook, or the last textbook. And again, like I said, they are incompletely dominant, but we still do use these dominant and recessive alleles. That is still part of the conversation. So as you can see in this case, that red flower has the dominant allele. The white flower has the recessive allele. So when you cross these two, actually, are there any predictions? I know I've mentioned some of this, but I haven't specifically mentioned this. But specifically, I did say the F1 generation. Well, no, I did. I guess I did give it away. Anyway, when you mix these two, you get a pink flower, right? Because no matter what, this thing's homozygous dominant, this thing's homozygous recessive. As you should know after lab, after having some experience with the Punnett squares, when you mix the two, 100% of the offsprings are going to be heterozygotes. And in this case, because we're dealing with incomplete dominance, the heterozygotes are a blend of the two phenotypes. Does anybody have any questions about that? It's pretty simple stuff. To me, this is the most simple. Because back before I took biology and knew anything about genetics, I honestly just thought this is the way everything worked. I honestly thought, all right, if you mix this thing with the mom and this thing with the dad, then the kid's going to come out with some sort of in between. I honestly thought that's how it worked. Kind of makes sense. It's intuitive. But again, this is not always the case, but this is the case of incomplete dominance. Here's the next question. Think about this in your brain, because you've done this before. And you've seen this before. Not the color. Just right for a second, just focus on the genotype. What we're going to do is cross these two, right? So we're going to take a pink flower. We're going to cross it with another pink flower, right? So we're crossing the F1 generations. So in your brain, if you can, try to picture what the Punnett square is going to look like. You're crossing two heterozygotes. So does anybody remember the phenotypic ratio for when you cross two heterozygotes? Because some of you did that in lab. All right, if it'll allow me to draw it, let's take a look. This is good too, because you need this experience. So here we go. Heterozygous up top, heterozygous at the bottom, or to the side, I guess. So we have one big R, one big R, little r, another big R, little r, and a little r. So what question, excuse me, what color is that one that I just circled? What color would we expect that flower to be? It's big R. What color would it be? Red. Good. Perfect. Because I already told you that. We already have one up on the screen, right? That one at the top left, it's red. It's big R, right? Here's another easy one for you. Little r. What color would that one be? White. Good, right? We can see that right here. And how about these two? These two heterozygotes, big R, little r. What color would they be? Pink. Good. All the information is already on there on the board, right? So there you go. And that's basically what we just wrote. So that's it. It's that simple. And again, you will have some questions where you have to do some Punnett square stuff. Mostly that'll deal with disorders, and it'll deal with things like complete dominancy, which is what we talked about previously. When we get questions about this, there's not going to be much math about it. It's going to be mostly things like, this thing mated with this thing. These were the results. Some of these offspring had this, some of these offspring had that. There might be some numbers involved in it. But in those kind of questions, you're not going to need to do a Punnett square necessarily. And you're not going to need to necessarily do any math. It's just, again, if I ever mentioned anything crossing, two parental things crossing, and the offspring having something in the middle, I should tell you it's incomplete dominance. Anyway, any questions about that? All right, so the example I gave, as you can see, is a flower. For independent work, you just look up some other examples that have nothing to do with flowers. Like, what about humans? What are some incomplete dominant examples for humans? Because who cares about hypothetical red and pink flowers, right? Well, you might, and no offense if you do. Anyway, here is one example. So you don't want to use this if you look it up for independent work, because here it is. It is just an example. So let me put it next to this, indicating I'm not going to ask you any questions about this, but I think it's a good example. Hypercholesterolemia, which basically means genetically you have really, really high blood pressure. And I mean really high. How many of you guys know people who have just high blood pressure? Maybe they take medication for, a few people, right? Like a statin or something. This is beyond that. This is really, really high. So, like dangerously high. What happens is if you're heterozygote, so again, this is what makes this a conversation about incomplete dominance. If you're heterozygote, you have high cholesterol. If you're homozygous, you have really high cholesterol, right? And I'll show you what I mean on the next slide. And again, these are just an example, so you don't need to know 2 times as high or 5 times as high, or the fact that somebody might have a heart attack or at age 2. These are just examples. I'm not going to ask anything about this. But it is a great example. This is from the old textbook. But what I like about it is not only is it a blend of phenotypes, which is what I'm trying to teach you, but it also, in this example, it shows you how that's working. Like, how do genes cause a blend of phenotypes? Either gene codes for this or it doesn't, right? In this case, this is how it works. So these are homozygous recessive, meaning those alleles, in this case, are just broken. So they still have the gene to make these little LDL receptors in the cell. They have the gene that says this is how you make it, and those are the things that take cholesterol out of the blood. But in this case, the allele that they have is broken. Does that make sense? Like, it still has the gene, but it's broken. So yeah, it has a set of instructions. It's supposed to say, hey, build these things, but there's something wrong with it, so it doesn't build it. So if they're homozygous recessive, they have none of those LDL receptors. Therefore, they don't take any cholesterol out of their bloodstream. Therefore, there's a lot of it in their bloodstream. Does that make sense? All right. The opposite is true of the homozygous dominant. That's a completely healthy person. Both of their alleles are dominant. And in this case, what that means is both of those alleles are functioning genes that say, hey, this is how you build this protein. And it works. So they build a lot of those proteins. So it takes the blood, takes the cholesterol out of the blood. And then, again, to the point of the incomplete dominance thing, this is the meat and potatoes of this discussion. Here we had a heterozygous person. They have the dominant allele and the recessive allele. So one of their alleles says, hey, this is how you build these LDL receptors. So it makes them, right? But the other allele is broken, so it doesn't have any instructions on how to make them. Therefore, if you have that, you make some of the LDL receptors, but not as much as you should. Therefore, you take some cholesterol out of the blood, but not as much as you should. Does all that make sense? All right. And this is the concept we're going to come back to later, too. I love this concept. Think about that in a few chapters. That'll be important. Think about how odd it is that you in your genome have genes that don't even work. In this case, only some people have that. But I'll go ahead and give you another example that we'll come back to later when we talk about evolution, because this is great evidence for evolution. Who knows why you have to? Well, no, I won't even say that. You guys know that you have to eat vitamin C, right? Vitamin C keeps you healthy. The reason is because you can't produce your own vitamin C. All mammals on Earth, other than primates, including humans, can make their own vitamin C. We're the only ones that have to eat our own vitamin C, or have to eat vitamin C because we can't make it. Why I find this interesting when it comes to evolution is, why would we even have the gene? Let me back up. We have the gene. There's a gene that says this is how you make the enzyme that makes vitamin C. We all humans have that gene. All primates have that gene. But all of our genes are broken. We have a broken gene. It's never worked for humans, as far as we can tell. So we just have a broken random gene in our genome. It's completely useless, but it's there in our genome. We have a lot of genes in our genome that are completely useless, and I find that fascinating. Why if you're building a human, why include the broken parts? But anyway, Any questions about incomplete dominance? Pretty simple concept until I do this. Then I'm going to make it trickier. Now there's something called co-dominance, and the blood groups are an example of it. So let me go ahead and tell you this too. Once again, the blood groups thing is just an example, just what I'm using to teach you about this type of dominance. I'm not going to ask you any questions about blood groups on the exam. Maybe for extra credit, but not for regular credit. There might be some questions about it on the study guide too, but not on the exam, not for regular credit. Anyway, I'm sure you guys have heard of blood groups. Who here knows your blood type? Just out of curiosity. I'm O positive. You know your blood type. He knows his. So if you don't, you might know your blood type but not know what it meant. That was me for the longest time. So now you're going to learn what it means a little bit. We're just going to scratch the surface. So it's called the ABO blood group, and it has 3 alleles of a single gene. And what I like about this is that not only now are we looking at a different kind of dominance, right? I'm calling it it's called co-dominance. So not only that, but now we're also looking at something else we've never seen before. So far, everything I've taught you has been 2 alleles. It's either this one or that one, right? Dominant or recessive. In this case, now all of a sudden I'm also throwing in a 3rd allele. So don't let that throw you off. And I'll go ahead and tell you now on the exam, this whole 3 allele thing will not be on there. It's good to think about because there are a lot of examples where it's not just 2 alleles. But for the exam, I'm going to focus on this whole idea of 2 alleles. So when you get a question on the exam for co-dominance, which is what I'm teaching you now, there's not going to be anything about 3 alleles. It'll be this thing mated with this thing. These were the results. This is an example of what kind of dominance, right? So anyway, that being said, keep that in the back of your mind as we talk about this. There are various combinations produced. So with these 3 alleles, that gives us 4 phenotypes. And those 4 phenotypes in this case happen to be blood types. You're either A, B, AB, or O, right? So those are 4 phenotypes that are resulting from 3 alleles. And as usual, well, not as usual, it used to be, remember, an uppercase was dominant and the lowercase was recessive. And this is still the case, except now we actually have 2 dominant things, only in this example, right? Because again, normally, or for the exam, when we talk about co-dominance, there's only going to be 2 alleles. But in this case, there happens to be 3. And of those 3, 2 of them are dominant, 1 of them is recessive. So I'll go ahead and tell you what they are. Basically, these genes, these alleles, code for carbohydrates that are on the outside of your blood cells. Not directly, because as you should know by now, a gene is in instructions on how to build a polypeptide or a protein. So long story short, what's really happening here is that those alleles code for a gene, there's code for an enzyme, and that enzyme helps make these carbohydrates. But we'll cut out the middleman and just, for the sake of the conversation, make it easier, just say that these genes code for carbohydrates on the outside of your cells. So let's take a look. So if you're group A, right, you could have any 3 of these genotypes. Again, this is just an example, so if you're taking notes, I don't know, I wouldn't even take notes for this part. Just soak it all in because it's just an example. I'm not going to ask you. Plus, again, we're dealing with 3 alleles. We normally don't do that. Anyway, to have the phenotype A, you can have any of 3 genotypes. You could have a big A. That looks weird. Oh, yeah, that is weird. Sorry, it got all bobbled up there. Should be big A, I big A, or I big A with a little I. In other words, the A is dominant, right? So as long as you have that A genotype, that A allele, as long as you have at least 1 of them, then you're going to produce that A carbohydrate. Hope that makes sense. Even if you have this recessive allele, that little lowercase I that doesn't say anything, it doesn't say, hey, these are the carbohydrates. The recessive one says don't make any carbohydrates. But even if you have that recessive one, the dominant one dominates over it. So you still have that carbohydrate A. So with B, right, you can either be homozygous dominant with the B, or again, you can be heterozygous where you have a big B and an I, right? But in this case, the I says don't build carbohydrates. It's ignored. So you still have that carbohydrate of B no matter what. In the case of AB, this is your genotype, right? You would have both the dominant A and the dominant B, right? So in this case, your blood cells produce both carbohydrates. And of course, if you're homozygous recessive, you're not producing any of the carbohydrates. So as far as our conversation is concerned, that's the most important because that's talking about alleles and genotypes and phenotypes. But just for your own knowledge, just in case you're wondering how that works medically, if you are blood type A, right, if you have carbohydrate A, your body does not like blood type B in it because it thinks it's not, well, it knows that it's not your blood because your blood has carbohydrate A. So when any carbohydrate B gets introduced into your blood or your body attacks it, right? So that's why if you are blood type A, only blood you can take is stuff with A in it, right? So you can have A, you can also have O because O has no carbohydrates. So it isn't so much that blood type A likes A, it's just that it hates B, right? So it cannot have B blood type. It can't even have AB blood type. So yeah, you've got the A, but you also have the B, and you can't have the B. You're anti-B. The opposite is true of the B blood type. You are anti-A. So obviously you can't have the A blood type. Obviously you can have the B blood type, but also you cannot have the AB blood type because even though it does have the B, you are anti-A, so you can't take it. And as always, O doesn't have any carbohydrates, so there's no problems there. AB blood type is the universal acceptor. It doesn't hate anybody. It's not anti-anything. Because it has A and B, it's like, take it all, take it all. You can give me, obviously, you can give me AB. I like it. You can give me B because I'm not anti-anything. You can give me A because I'm not anti-anything. You can give me O because it has nothing. And then, of course, you have O. O is what we call the universal donor because, again, as I've just got finished demonstrating to you, nothing is anti-O because O doesn't produce any carbohydrates. Whew. Any questions about that? All right, now that I've given you that example, let me give you some easy examples that you're going to see, not these specific examples, but this is the level of easiness that you're going to see on the exam. Forget all that. Here we go. Codominance. Here's what you need to know. So again, let's take a step back and talk about what we've already learned. Complete dominance, which is the first thing you learned. You had this thing was dominant to this thing, right? So if you mix them, the dominant thing always showed up. Then we talked about incomplete dominance, which is the pink flower example, which is where the two phenotypes blend, right? The offspring are a true blend of the two. This one is similar, but slightly different. Now, instead of blending the two phenotypes, you just see both of the phenotypes individually, right? So if you mix a red flower and a white flower in this situation in codominance, then you get a red flower with white spots or a white flower with red spots. Does that make sense? It doesn't blend like it does in incomplete dominance. They both hold their own individual identities. Or as these other examples written on the board here, you have a black dog and a white dog, and then you have black dogs with white stripes or white dogs with black stripes. That would be an example of codominance. Hypothetical. It's actually a lot more complicated than that when it comes to dog colors, but these are just visualizations for you to think of. A red cow and a white cow gives you a white cow with red spots. That's actually a real example. That's not hypothetical. And again, the example I just used with the flowers. So again, for the exam, I'm going to give you some random example. I'm probably going to make up something. It's going to be completely fake. I'm going to make it up, but that doesn't matter. The thing is, you should recognize if I say this offspring mated with this offspring, and then this parent mated with this parent, these were the offspring, and they had both phenotypes, not a blend of the two, but both individually, then we'll say that is codominance. Anybody have any questions about that? All right. To me, that's where it got more complicated. Only because codominance and incomplete dominance, they sound similar phonetically, and then the situation is also kind of similar. So to me, the hardest part was always remembering the difference between the two. And my best advice is just read as much as you can, study as much as you can, watch as many little videos as you can to help you remember. All right. So let's talk about pleiotropy. This is where it gets a little bit more complicated. And as a matter of fact, most of the questions when I say this thing mated with this thing, these were the results. Most of those questions are either going to be complete dominance, incomplete dominance, or codominance, most of them. This is going to be a sort of different situation, and you'll see what I mean. Pleiotropy. Let's talk about it. In this case, instead of having one gene controlling one character, like we've talked about, so far everything we've talked about has been one gene, one character, even if it was the blood groups. Sure, there was 3 alleles, but there was one gene controlling the blood group, the blood type. Here we have one gene that controls several characters. So you need to know that for the exam. Sickle will be the next word for attendance. And also, let me put a big old X through this. Sickle cell is just an example. There might be questions about it on the study guide. I might give you some extra credit questions about it, but there will not be any regular questions about sickle cell for this. This is just an example. Actually, we'll talk about sickle cell later, twice. We're going to talk about it in the concept or the context of mutations, and then we're also going to talk about it in the context of evolution. You might get some questions about that, but not this. Anyway, sickle cell disease is an example of one of those things that exhibits pleiotropy. What does it do? It causes abnormal hemoglobin proteins. Those are the proteins in your blood. It causes your red blood cells to get a weird shape, as you can see at the top right. That's the weird shape. That's what it looks like when it's not healthy, when it has sickle cell disease. And it's usually only the people who are homozygotes that have this disease. Not that you need to know that either. Again, just an example. So here you go, an individual who's homozygous for that sickle cell allele. So again, they have both of those alleles. Therefore, they have sickle cell. Therefore, they have the abnormal shaped hemoglobin. And that abnormal hemoglobin crystallizes into these long flexible chains, which then causes a bunch of other stuff, right? Weakness, pain, organ damage, paralysis, all this thing from one gene. And that's the long story short. That's what you need to know for the exam. When we talk about pleiotropy, it's one gene causing, well, let's just say many different characters, or excuse me, more than one character. Even technically two would be more than one character, right? So that would count as pleiotropy. And again, the example that I'm going to give you, I don't even know what it's going to be yet. It's going to be off the wall. It's going to be hypothetical. It's going to be fake, and you need to recognize it. So I might say something like, I don't know, you're homozygous for a gene on your third chromosome. Your dad had the recessive alleles. Your mom had the recessive alleles. You have the recessive alleles. And this gene has caused you to have hairy nipples, bad breath, and oozy ears, right? Weird off-the-wall example. And you're like, oh, I know right off the bat that's pleiotropy because he just said that's one gene, and it's causing these three things that have nothing to do with each other. Hairy nipples, bad breath, and oozy ears, right? But anyway, any questions about that? Or, I mean, even though I'm not going to test you on it, are there any questions about sickle cell disease? All right, nice and easy. Now we'll talk about polygenetic inheritance. I need to fix that. Every semester I see that. Every semester in the lecture I say, I need to fix that. And then I forget to do it. But maybe this will be the semester I think to fix it. Anyway, it's not polygenetic inheritance. It's polygenic inheritance, like that. In a sense, polygenic inheritance is the opposite of pleiotropy. So remember, we just talked about pleiotropy, which was one gene controlling a lot of different characters. Now we have one character controlled by a lot of different genes. Or as the book puts it, it is the additive effect of two or more genes or of a single phenotype. So once again, remember, I'm not going to ask you the definition of polygenic inheritance. I'm going to say this thing mated with this thing. These were the results. What is this an example of? Right? Or I might ask you this way, because this is another thing. Of all the things that I've taught you, all these different ones, this is the only one that would give you a bell curve. And let me give you an example before I show you the next slide. And this is why you had to do this in lab. Oh, come on, please. Don't do me like that. There we go. If you were to chart something online, right? Not online, in lab. Let's say you did the whole human population, and you were gonna chart the whole human population, who has a dimpled chin and who has a smooth chin. It might look something like that. Without that stupid sidelines. Try to imagine without the little zigzags out there. But can you imagine that bar graph? It would have two bars. Does that make sense? You're either dimple chin or you're not. That's not a bell curve. But a bell curve is, in a sense, a bar graph that's just been someone like drew a line around it. See what I mean? That's also a bar graph. So if you were to bar graph this situation, which is height, and even this is oversimplified. Height is not this simple, but it's a good example of a way to visualize it. Here, we're looking at three different genes controlling one character. And again, height is just an example, and this is not how it works. This is just to visualize things. Right? So if you were to plot everyone's height, this is what it would look like in this simple situation. Now, in real life, too, though, if you were to plot everyone's height and not use the genotypes like we've done here, the fraction of the population, if you were to do like the height, like their actual height, how tall are they? Oh, they kind of did that. It would look like this. Forget the bars, but it would have a graph, right? The average human, I think, is like 5'6" or something around there. There's a lot of people who are average, right? But there's even fewer people who are tall, even fewer people who are really, really tall. Make sense? Same here with the shortness. There's a fewer people who are short, even fewer people who are really short, even fewer people who are really, really short. So that's what I mean by it's on a bar graph. So that is definitely one of the ways you could look at that for the exam. That might be the key to that answer. Anything that you could plot on a curve. I'm sorry, I said bar graph. It is a bar graph. But specifically, if it has a bell curve, then that means that it's pleiotropy. Because again, if you're just plotting dimpled chin versus smooth chin, that bar graph is only going to have 2 bars. So every time you add another gene to the equation, that's more bars. And again, height actually has a lot more. It's more complicated than this. But this is only 3 genes. So this is a Punnett square, by the way. This is a lot bigger. The Punnett squares you guys have done have always been with 1 gene, right? So it's either this allele or that allele. Now we're talking about 3 genes. It's still a Punnett square. And also, too, keep in mind, that's only 3 genes. Imagine if we dealt with more genes. Or if it was something like the blood group. So like not only are we dealing with more genes, but what happens if there's more than 2 alleles? But anyway, any questions about pleiotropy? And again, like I mentioned earlier, these are all great independent work topics. I've given you examples of complete dominance. I've given you examples of incomplete dominance. I've given you examples of codominance. I've given you examples of pleiotropy and now polygenic inheritance. But what you can do for independent work if you want is look up some other examples. Human examples if you want, because that's obviously more relevant than flower color or cows. Anyway, any questions about polygenic inheritance before we talk about this last category, which is epigenetics? All right, this one really throws a wrench in everything we've learned. So everything we've said, this just makes it completely more complicated. Good grief, I can't think today. Many characters result from the combination of heredity and environment, right? So everything we've said thus far, at least the way we've described it, has been an example of the genes control this character. You have these genes, you have this character. But now what we're saying is that's not always the case. A lot of the times, the environment controls the genes. So let me use this analogy, and I'll use it again later in the semester. So now I think the first time I use this analogy. Imagine your chromosomes are books, right? You've got 23 pairs of books, and in those books are instructions on how to build proteins, right? Because that's what a gene is, right? So instructions on how to build proteins. What we're saying here is that sometimes some pages are easier to get to than others. So if you want to build a protein, sometimes it takes a lot of work to get to that page. Maybe the pages are stuck together. I don't know, maybe the page is real flimsy, whatever the case may be. And sometimes, you ever had a book that you read so much, or you kept it upside down, and when you leave it open or leave it out, it just kind of opens up and just stays open? You ever had a book like that? Like you've abused the book, so it just lays open. You can't really close it. It just flops back open. You ever had that? In a sense, that's sort of like your genome, right? Sometimes there's things you can do to your book where it's just wide open in that one page, right? That one, every time you open that book, it just kind of opens to that page. Therefore, the information in that book or on that page is going to be read more. Same thing here. There's things going on in your genome that we'll talk about later to where sometimes the instructions on how to build a protein, those particular instructions are really hard to read, so they aren't read much. Sometimes they're really easy to read, so they're read a lot. That's the analogy. But also on a less hypothetical way of describing it is sometimes genes are turned on and genes are turned off by what's happening in your environment. And that is called epigenetics. That's what you need to know. That main bullet point right there, that top one. Yeah, we'll talk about these other ones too, but this is just to give you some context of why this is important. Whether some things are more influenced by genes or environment or nature versus nurture is a very old and hotly contested issue. And you can write about that for independent work if you want to. Try to keep your opinions out of it. Like I would love to have you write an objective paper about something that's a hotly contested issue, like homosexuality, right? They say is it genetic? Is it nature? Is it nurture? More likely, is it some sort of mixture of the two, which would be epigenetics, right? So if you want to, think of anything you want and write a paper about it. And again, I would recommend you being objective. I don't really care about what your opinion is. You could just say, some people think this, some people think this. Here's the evidence that supports that. Here's the evidence that supports that. Anyway, a good example of that, and again, this is just an example. If you spend any time with identical twins, like a lot of time with identical twins, and you can tell the difference between one and the other, this is a great example. They have the same exact genes, right? Their books of instructions on how to build proteins are the same exact things. So how is it that they could be a little bit different, right? Well, just because they have the same exact instructions on how to build proteins doesn't mean they're using all those instructions all the time, right? So twin A might build this protein right now. Twin B might build some other protein. How many of you have spent know any identical twins well enough where you can tell the difference between the two? A few of you, right? I haven't had I've met some, but I don't know any that well. But it really is a good example. And not just the personality too, but sometimes there are things like when you see them enough, you can look at them and say, oh yeah, that's Bob, that's Frank, right? You can tell by looking. And that's why, right? Because of epigenetics. Yes, they have the same genes that result in the same proteins being built, but genes need to be turned on and genes need to be turned off. And the environment is part of what turns them on and off. So even these twins who are usually in the same environment, but not completely, even then, they are slightly different because their genes are getting turned on and turned off differently than each other. This is also something I'm not going to test you on, but it is worth noting. This will be really important in the future, I think, once science knows a little bit more than it does. But in general, it's only the genetic influences that are inherited and the effects of the environment are not passed on, right? So in general, another way of what that's saying is when you have kids, it's usually just your genes that you're passing on, right? It's your alleles that you're giving to your offspring. But recently we found out it's not just your genes, it's also this epigenetics thing, right? Something that the environment is doing to our chromosomes, to our books, that's causing them to stay open to that page or causing this page to stick to that page, using this example, right? So there's things that we are doing in our lives, the environments are doing to us, that's not changing the genetic code, the words in the book, so to speak, but it's changing the book itself, right? So it changes how those genes are read. And we recently found out that some of that is heritable, which is scary in some ways. Because I think one of the examples, and you can look this up for independent work if you want, is like cigarette smoking. So with cigarette smoking, usually the way it gives you cancer, one of the ways it gives you cancer is it mutates your DNA. And as you know from the mitosis discussion, if you mutate any gene that's involved with your cell cycle, that might result in cancer. Anyway, another thing cigarette smoking does, other than just mutating your DNA sequence, is also just changes your book, so to speak, changes the chromosomes, not the code, not the letters, but the chromosome itself. And unfortunately, if I remember correctly, that is one of those things where once you pass that along to your offspring, you can also pass that along to your offspring. That's the bad news. The good news is, though, for some of these things, that's also reversible. Because if it's environmental and you're doing something to your chromosome that's messing it up, but it's not changing the code, and sometimes if you stop doing that thing that's messing up your chromosomes, then they'll fix themselves. Yeah, and that's what that is right there. That's basically what I said right there. You can read that. The point is, not only can you change the sequence of the DNA, which is all the stuff we've been talking about, the sequence of the DNA, your genes. Not only can you do that, but you can also just change the chromosome itself, the mechanisms, the things that read your DNA. Anyway, just an example. And again, like I said, that's kind of scary in one way because the things you're doing to yourself are not just to yourself. You could also pass that down to your offspring. That's the bad news. But the good news is in some cases they are reversible. Anyway, any questions about all that? Ooh, this we can go through quickly. So now we're going to talk about the chromosomal basis of inheritance. And I've already been doing this technically. So the way the textbook presented this information, it just talked about this like, hey, these genes, these alleles, blah, blah, blah. But what I've been doing, and your book hasn't been doing, is instead of just saying your genes and alleles, I have been talking about your chromosomes. So your book now goes back and says the same thing that it said earlier, but now it's saying it in the context of the chromosomes instead of just the specific genes. So again, I basically already said all this, not all of this, some of this. We're just going to say it again. So the chromosome theory of inheritance states that genes are located at specific positions on chromosomes. You already know that. If you're taking notes, there's no need to write that down. We said that on Monday, I think, right? You know that genes are located at specific positions on specific chromosomes. Who did the paper? I don't know if I expect you to remember it, but did anybody do the paper on the P53 gene and happen to remember which chromosome it's on? Do you remember which chromosome it's on? There you go. Right. So, and I don't know if he's right or not, but let's say it is. Okay. He's going to look it up. But P53 is a gene, right? It's a very important gene. And let's say it's on chromosome number 13 in his body. Chromosome number 13, I don't know, right towards the tip. That means for every single human on Earth who doesn't have something wrong with them, on chromosome number 17, in the same spot is that gene, right? So this is what we know. That is the chromosomal theory of inheritance, right? That's what makes all humans, one of the things that makes all humans connected. For the most part, every gene we all have it at the same chromosome at the same location. And as you already know from the previous chapter, the behavior of the chromosomes during meiosis and during fertilization accounts for all these inheritance patterns that we're talking about, right? So all these complete dominance, incomplete dominance, pleiotropy, all this stuff we've been talking about, it can all be accounted for by all the stuff that you learned in the last chapter, right? So it's the chromosomes that undergo the segregation. It's the chromosomes that undergo the independent assortment. It's the chromosomes that account for Mendel's laws that we've been learning about in this chapter. And this is all that your book is trying to do here. It's kind of combined the last chapter with this chapter. And like I said, I feel like I've been doing this throughout the chapter, so we can go through this quickly. So here we go. Now we're looking, again, we've already looked at this. We used this example. The first time I told you about a dihybrid cross, we were talking about the pea plants. And I said, all right, we're looking at, instead of one character, we're going to look at two characters, which is the color of the seeds and the smoothness of the seeds, right? So you can either be yellow or green, or you could be, and you could either be smooth or wrinkled, right? So we're looking at two different characters. And I'm not even going to spend too much time on this because we've already talked about it. The point is, see, that's really confusing. You can study that if you want. There's nothing to memorize here. There's a lot of information. The point is that these alleles are on chromosomes, and it's the chromosomes that line up in meiosis, and it's the chromosomes that get separated in meiosis. So every time I've been saying the allele does this and the allele does that, what I really meant, and I actually have been saying it, is that it's the chromosomes that are doing all this, right? I'll try to find a good video on YouTube to share with you. One that makes it a little bit less confusing than this garbage. Anyway, that being said, let's talk about linked genes. There's not going to be hardly any questions about this on the exam, so we're going to go through this quickly as well. Linked genes are located near each other on the same chromosome, right? So gene 2 and gene 3 are really linked with each other. Also a little bit, they're linked to gene 4. Also a little bit gene 1. Let me give a really quick example. Let's assume that this is a chromosome from your mom, and this is really hypothetical. I'm going to stick with some of the stuff I've already been saying. Let's say gene 2 is for, I don't know, hairy nipples. Hairy nipples. God, I hate this thing so much. That's supposed to be an HN. Anyway, gene 2 is for hairy nipples. Your mom's got hairy nipples. Sorry to tell you. Gene 3 is also for smelly breath. Well, depending on the allele, right? So you either have smelly breath or you don't. Or just like for gene 2, you either have hairy nipples or you don't. In this case, sorry to tell you, in addition to hairy nipples, your mom also has smelly breath. Gene 4 is for, I don't know, hairy soles of the feet. So if you have it, you've got hairy soles. If you don't, you've got regular smooth soles. So once again, I'm sorry to tell you, but your mom has hairy nipples, smelly breath, and hairy soles on her feet. Now, the point I'm trying to make here is that's her chromosome, right? So now imagine if you could, this is probably not, God, I would kick this thing. You ever seen Office Space where they take that thing out in the field and they just beat that piece of electronics equipment? I just want to beat this computer sometimes. Anyway, that's supposed to be your dad's gene. Now imagine your dad has the opposite. So he has bald nipples. He's got nice breath, and he has regular old bald bottom of the feet, right? That's your dad. What this is saying, if they're linked genes, right? If you inherit your mom's hairy nipples, you're probably also going to get her stinky breath. Does that make sense? Because they're on the same chromosome. It's not like when we go through meiosis, like this allele goes this way, this allele goes that way. No, the chromosomes are going. You either get your mom's chromosome or your dad's chromosome, right? Of course, there's also crossing over. So there is a chance you get like your mom's chromosome with a chunk of your dad, in which case, in some cases, you might get the hairy nipples and the smelly breath, but have bald feet. Does that make sense? What I'm saying, linked genes, they're on the same chromosome. Therefore, unlike everything else I've ever taught you, sometimes one thing does lead to another. So in this hypothetical situation, if you have hairy nipples, if you got your mom's hairy nipples, you're probably going to get her smelly breath. Does that make sense? Because they go together. And again, that's why you get something other than a 9-to-3 to 3-to-1 ratio. If those two things were together, then that means every time you had the hairy nipples, you would also have smelly breath, and you would just get a regular old 3-to-1 ratio. And the last thing I want to say about it, because it's on the study guide, is there's actually mathematical ways to do this. You can actually look at crosses and look at ratios and figure out if a gene's linked, and if so, how closely it is linked to each other. I think your textbook might even describe how to do that. So if you're taking notes, write this down. You can learn about it if you want, but I'm not going to test you on it. I think it's complicated. I think as a non-biology majors, especially freshman non-biology majors, you're not going to need to know that. But just know that linked genes are close together. And if you want to understand how it works, read it, and I'll answer your questions privately. Not that it's a secret, but I just don't want to bother anyone else with it. The next word for attendance, I'm pointing at it right there. So again, linked genes, they're close to each other on the chromosomes. That way, if you get one, you probably get the other. And obviously, again, the closer they are, the more likely that you are to get them. If they're farther apart, there is a higher chance that crossing over will happen. So you might get your mom's chromosome, but you might still get an allele from your dad because of crossing over. Whew. This one's really easy. Sex determination in humans. We've already talked about this. We can go through this very quickly. Many animals, including mammals, have a pair of sex chromosomes designated X and Y. You need to know this for the exam. Males are X and Y, females are X and X. We already talked about the exceptions to that rule in meiosis. Sometimes there's extra X's, sometimes there are extra Y's, sometimes there's missing X's. There's all kinds of different exceptions to the rule, but generally that's how humans work. Speaking of which, if you ever hear anybody say some garbage about biologist, what is some stupid thing I saw? Something about the vaccine, like the same people who are telling you there's more than two sexes are telling you this vaccine's safe. Well, those are completely two different things. First of all, we're talking about there are no two different sexes. There's only two sexes, male or female, for humans. But the thing people are bitching and whining about right now, pardon my French, is gender. Gender is completely different than sex. If you want to have a conversation about gender, you would talk to somebody who knows things about social science, which is not me, right? Like psychology, sociology. That's that wheelhouse. I'm not saying anything good or bad about it. I'm just telling you I don't know anything about it. I'm a biologist. To me, there's male or female. I understand that gender is a spectrum and all that. I know a lot of people who identify a bunch of different ways. I love them, don't care. But when it comes to sex, this is the only reason I'm saying this is not to point out this is what sex is. I just want to make sure everyone knows biology and social science are completely different. But I hate it when people say things like that about biology. It's like, you probably don't even know the sexes. I'm like, no, the sexes are pretty simple. I know that. The genders, on the other hand, that's more complicated. They're completely different things. Anyway, for the exam, know that. Males are XY, females are XX. For independent work, you could look up the exceptions to that rule. Sex is a very interesting thing. Some things are hermaphroditic. I just posted something on my Facebook where there's different types of flatworms that they're both male and female, and the way they mate is they try to poke each other with a penis, and whoever pokes the other worm first, that is then the male, and the one who got stabbed by the penis is then the female and becomes impregnated. It's all so fascinating. Anyway, there's the X chromosome, there's the Y chromosome. When we get back on Monday, we will finish this chapter and jump into the next one. And I guess the last word for attendance will be Saturday, because it's almost Saturday. You guys have a good weekend. Keep an eye on your grades in My Online. I'll try to update them if it doesn't do it automatically. The exam is now past due. Well, in 10 minutes it's past due. So I will put the grades out there. That's it. Have a good one. You too. And if you guys need I was about to say, if you need anything filled out, just come with me to my office.